This video outlines the soil and root processing of samples from Pinus radiata for DNA extractions. Returning from the field, these samples need to be processed in the laboratory. The first step involves homogenizing the sample by thoroughly mixing it. After homogenizing, subsample 1/4 of the sample into another clean bag to be used for soil and root DNA extractions. Process the rest of the remaining soil through the soil processing standard operating procedure. Returning to the smallest subsample, sieve the soil using a 2 mm sieve to separate the roots and soil. Carefully remove some of the roots from the sieve. The sieved soil is now ready to be used for soil DNA extractions. Use the QIAGEN DNeasy Power Soil Kit for both soil and root DNA extractions. Use aseptic techniques and ethanol to sterilize equipment to keep samples free of contamination. From the sieved soil, add around 250 mg of soil per sample to 4 separate power bead tubes. These tubes are now ready to proceed with DNA extractions. Moving on to the root DNA extractions, place the roots into a calcium chloride solution for washing off the excess soil. To help remove the excess soil, place the tubes in a shaker at 150 RPM for 1 hour. If the roots still have lots of adhered soil, refill the tube with fresh calcium chloride solution and wash for a further hour. Carefully remove the washed roots and cut these up with scissors into small pieces to create a homogeneous sample. Add some of these cut-up roots into 4 power bead tubes per sample. These are now ready for DNA extraction. Homogenize both root and soil tubes through mechanical processing before proceeding with extractions using the manufacturer's instructions. Elute the final DNA into 80 microliters of C6 solution. From each tree there will be 12 DNA samples with 4 each from soil, root and needles.